Imagine starting your day with a ward full of complicated UTIs, and before your shift ends, each one has moved from empiric uncertainty to a confirmed pathogen and antimicrobial resistance profile. This paper shows that timeline has finally collapsed to hours.
That is the operational dream, isn't it? And that's exactly what this new paper in Nature Communications is really putting on the table. And what they've developed, this unified metagenomic method, it really does feel like a genuine paradigm shift.
It does, because usually when I hear metagenomics, my brain goes straight to, a last resort
The
test.
ICU patient, right? Nothing's growing,
Exactly. We're desperate. But this team, they're pitching something completely different, a frontline screening tool for just routine urine
which
samples.
is, it's ambitious. And the headline is all about speed. They've managed to get this whole workflow down to about 4 hours.
4 hours.
Think about the standard. You send the culture, you're waiting 2, maybe 4 days.
hmm
With this, you're not even home for dinner, and you have a pathogen ID and a resistance profile.
Okay, that's a massive difference. So let's walk through how they actually pulled that off. Where did they find the time savings?
Well, they made a pretty controversial choice right from the get-go.
Which was?
They completely eliminated PCR preamplification.
Oh, wow. Okay, that sounds risky. I mean, don't you need PCR to get enough DNA to even sequence?
You usually do, yes.
Okay.
But PCR takes time, about 3 hours right there. So skipping it is an instant time save. But there's a bigger scientific reason.
Okay.
PCR has bias. It amplifies some DNA sequences more than others. It distorts the picture. By getting rid of that step, they preserve the full resistum.
Right, the resistum. Help me out with that term. We hear it all the time, but what are we actually looking at?
So think of it as the bacteria's entire arsenal of weapons. You're not just IDing the bug. You're seeing every gene it has to fight off our antibiotics.
Mm-hmm.
If you use PCR, you might accidentally hide some of those weapons or make others look bigger than they really are. Skipping PCR gives you an honest, unbiased look.
Okay, so you get a truer picture. But if you don't amplify the bacterial DNA, then what about the needle in a haystack problem? I mean, urine is just full of human DNA. So you need an enzyme that can actually work in those conditions.
You do. And that's where specific enzyme chemistry comes in. The study used artaxymes MSAN and H-alisan. These are salt-active nucleases.
They're evolved for high salinity environments, right?
Exactly. They don't just tolerate the buffers, they thrive in them. Because of that, the researchers achieved something like a 1,000-fold depletion of host DNA.
That's the game changer. You've removed the haystack, so the sequencer only sees the needles.
And that efficiency drives the cost down. That is the classic bottleneck. In a typical sample, the human DNA it completely overwhelms the bacterial signal.
So you'd just be sequencing the patient.
Exactly.
Yeah.
To make this work without PCR, you absolutely have to get rid of that human DNA first.
And how did they manage that? How do you scrub the sample clean without, killing the bacteria you're looking for?
So it can just chew up all that host DNA right there in the buffer, leaving the bacterial DNA untouched.
But here's the part of the paper that really blew my mind. They didn't just build a faster test. They sort of changed the whole logic of the test itself.
You're talking about the screening strategy.
Mm-hmm.
And yes, that is the core innovation here. It's the thing that makes it all economically viable.
The insight being, don't sequence everything.
Don't sequence everything. They realized a lot of urine samples sent to the lab are actually negative. So they put in a triage step.
A filter.
A filter, exactly. Before any sequencing, they just count how much bacterial DNA is in there. They use flow cytometry or measure the DNA yield, and they set a hard threshold. I think it was anything above 451 bacterial cells.
And if a sample is below that number?
They just stop. They assume it's negative or contamination. They don't waste time or money sequencing it.
Which, okay, that has to bring us to the cost. Metagenomics has this reputation for being, $1,000 test.
Not here. Because of that triage, they figure the cost to just screen a sample is about $6. And if it's positive and goes on to full sequencing, it costs around $36 total.
$36. That's cheaper than a lot of commercial kits just for the prep.
It's a game changer. That price point is what lets you even consider moving this out of the ICU and into routine use.
But none of that matters if the results are wrong. How is the accuracy?
The stats were really impressive. 99% accuracy for identifying the pathogen. And for the antimicrobial susceptibility, which drugs will actually work, they hit 90% agreement with the standard methods.
Did they find anything that the standard cultures missed?
They did, and this is important. They found things like Actinidignum scaliae and Aerococcus urinae. These are bugs that are notoriously tricky to grow in a lab, so they often get missed, but they cause real infections.
So putting it all together, a clinician can order this in the morning, screen it for $6, and if it's a real infection, have a full resistance profile back before their shift ends.
Yeah. It shifts the whole model from being reactive to being proactive. You stop guessing with broad-spectrum antibiotics for days and
Yeah.
have actual actionable intelligence in hours.
It feels like it finally closes the loop. This is what we've been wanting for so long.
It really does. And if you take that to its logical conclusion, if diagnostic stewardship can be this fast and this affordable, it poses a huge question.
Which is?
Could this be the beginning of the end for empirical prescribing in complicated UTIs?
Meaning we stop guessing entirely.
Why would you guess?
Huh.
If you can know the answer for $36 in 4 hours, there's no clinical reason to guess anymore. And that changes everything.
That is a fascinating future to think about. Huge thanks to the authors for this work. And of course, thank you for listening.
Until next time.